All right, let's dive into one of the most fundamental techniques in all of biology. It's a process that's a total game changer for everything from diagnosing diseases to creating new medicines. And the best way to think about it, it's like a microscopic marathon, a great protein race where thousands of tiny molecules compete, and the finish line reveals some amazing secrets about our own cells. So let's get to the starting line and see how this whole thing is run.
So just imagine this for a second. You're a scientist, and you've got this little tube. Inside is just a chaotic soup of thousands of different proteins, all with different shapes, different sizes, different charges. It's a mess. How in the world do you even begin to sort all that out? How do you figure out what you're actually looking at?
Well, the answer is actually pretty genius. Scientists figured out how to create a molecular obstacle course. They line up all the proteins and basically make them race through it. The technique is called SDS-PAGE. I know, it's a mouthful. It stands for sodium Dodecyl sulfate-Polyacrylamide gel electrophoresis. But trust me, by the end of this, that crazy name is going to make perfect sense.
Now, any good race needs a level playing field, right? You can't have some racers showing up on bikes while everyone else is on foot. It wouldn't be fair. So the very first, and maybe the most critical step, is getting every single protein prepped and ready for a fair competition.
See, the whole point of this race, the only point, is to sort proteins by one thing and one thing only: their size, their molecular weight. And to do that, you absolutely have to get rid of any other differences that could mess up their speed. That means taking all their unique, folded-up 3D shapes and stretching them out into straight lines. And it also means giving every single one of them the exact same electrical charge.
So, how do we pull that off? It really comes down to a chemical 1-2 punch. We use a couple of key ingredients that act like the race officials, making sure every protein is ready to go at the starting line.
Okay, first up, we have SDS. The easiest way to think of it is as a really strong soap. It does two crucial jobs. First, it breaks all the weak bonds that hold a protein in its folded shape, forcing it to unravel into a long line. Then, it completely coats that protein line with a uniform negative charge. Next, we bring in a reducing agent. This stuff is like a pair of molecular bolt cutters. It goes after the really strong, stubborn connections called disulfide bonds and snaps them. So to recap, SDS takes care of the shape and the charge, and the reducing agent breaks the toughest links.
In the lab, this is actually pretty straightforward. You just take your protein sample and mix it with a special loading buffer that's got both the SDS and the reducing agent in it. Then, you boil the whole thing for about 5 minutes. That blast of heat just makes sure the chemicals can do their job perfectly, leaving every protein completely unfolded, stretched out, and ready to race based on its size alone.
All right, our racers are prepped and ready to go, so now it's time to talk about the racetrack itself. And this isn't some flat, simple track. Oh no, this is a complex, three-dimensional maze that the proteins have to navigate.
The racetrack is this squishy gel made of something called polyacrylamide. You can basically think of it like a sponge or a really, really fine mesh. Now remember how we coated all our proteins in a negative charge? Well, we apply an electrical field across the gel, positive at the bottom, negative at the top, and those proteins start getting pulled right through it. The race is on.
And this is where the magic happens. That gel mesh is the obstacle course. The small, nimble proteins, they can zip through the pores pretty easily, so they move down the gel fast. But the big, bulky proteins, they get tangled up, slowed way down by the mesh. It's a race where size is literally the only thing that matters. The smaller you are, the faster you run.
Okay, that makes sense, but wait a minute. What if you're trying to separate a bunch of really tiny proteins? Or what if you're trying to separate some absolute giants? You wouldn't use the same obstacle course for a kindergarten fun run that you'd use for a pro athlete competition, right? Scientists have to be able to adjust the difficulty. And the way they do it is actually super clever and simple. It all comes down to the concentration of acrylamide in the gel. If you look at this chart, you can see how it works. A high percentage gel, say 20%, creates a very dense mesh with tiny, tiny pores. It's perfect for separating small proteins. But if you're trying to resolve the big guys, use a low percentage gel, which has much larger pores for them to move through. You just pick the course that fits your racers.
But here's a really, really smart part of the whole process. To make sure the race is perfectly fair, scientists almost always use a two-part gel. At the very top, there's a stacking gel. It's a low-concentration gel with huge pores, and its only job is to get all the proteins from your sample squished together into one single razor-thin starting line. Once they're all perfectly lined up, they hit the resolving gel underneath. That's the gel with the right-sized pores we just talked about. And that's where the real race, the actual separation, happens.
So the race is over, the power is turned off, the proteins have all separated out based on their size, but we've got a problem, a big one. We can't see them. The finish line and all the racers are completely invisible.
Yep, that's right. The proteins are colorless and the gel itself is transparent. It's kind of like having a picture on an old roll of undeveloped film. If you want to see the results of our race, you've got to add a dye, a stain that'll stick to the proteins and finally make them visible.
One of the most common ways to do this uses a dye called Kumasi blue. It's a simple three-step process. First, you fix the proteins, which basically just locks them into place inside the gel so they can't float away. Second, you stain. You just soak the whole gel in the blue dye solution, and at this point, everything turns blue. Finally, you destain. You wash the gel, and all the extra dye that isn't stuck to a protein gets washed away. And what you're left with are these beautiful, sharp blue bands, each one marking where a group of proteins finished the race.
And this is what it looks like in the end. Pretty cool, right? Each of those vertical lanes is a different sample that was loaded at the top. The proteins race down towards the bottom, and every dark band you see represents a huge collection of identical protein molecules that all traveled the same distance because they're all the same size.
Okay, so we've got these awesome-looking blue bands, but what do they actually tell us? How do we go from these pretty lines on a gel to real, useful scientific data?
I mean, seeing the bands is great, but that's just a relative position, right? It tells us this one's bigger than that one. But how do we know the exact size, the actual molecular weight of the protein in a band? We need some kind of ruler to measure them against.
And that's exactly what scientists use. It's called a molecular weight marker, or sometimes just a protein ladder. It's basically a pre-made cocktail of different proteins, and the key is their sizes are already known. You run this ladder in one of the lanes right next to your mystery samples. It's like having a few pace cars in the race where you know their exact speeds.
So check this out. On the left, you can see a real gel. See that lane with all the different colored bands? That's our ladder, our molecular ruler. Because we know the exact size of the protein in each one of those colored bands, we can use their positions to create a standard curve, basically a graph just like the one you see on the right. Now, all we have to do is measure how far our unknown protein band traveled, find that spot on our curve, and we can read its exact size. It's just a beautifully elegant way to get a super precise measurement.
So what's the final story the gel tells us? Well, if you see a single, really sharp band, that's a good sign you probably have a very pure sample of just one type of protein. If you see a bunch of bands, it could mean your sample is contaminated with other proteins. Or, and this is really interesting, if your original protein was a big complex made of several smaller pieces, those multiple bands could actually be showing you all the individual subunits after we broke them apart at the start.
So SDS-PAGE is this incredible, powerful tool. It lets us separate proteins, figure out their size, and check how pure a sample is. But it leaves us with one last, massive question. We've sorted the entire molecular crowd by height, but in that crowd of thousands, how do you find the one specific face you're actually searching for? Well, that's a whole other race and a story for another time.